Good afternoon. Can you hear me? Hello. Can you hear me? Hello. Oh, okay. Can you hear me? Okay. So in the morning we have completed with the gastric secretion along with bile. Actually, these are the two questions which have been asked in the physiology. A regulation part comparatively ask less to your your faculty. They are not giving more weightage to that. Only the part with composition, functions and importance related with that, which has been covered. So the part which is remaining now in the gastric that I want to cover today still students are joining. Somehow they have good problem with the, I think, range also. Okay, so we are starting with the pancreatic secretion today. Pancreatic secretion. This is also most important topic. Can you visualize my screen? Hello, no one giving the response. Can you visualize my screen? Hello students. At least say yes. Okay, thank you. Okay, it will. Okay, just to see whether you are visualizing my screen or not. Otherwise, I will continue, and afterwards you will say that, sir, your screen is not visualized. Then problem. Okay. So pancreatic secretion. What actually we need to learn in this? The digestive enzyme, secretion of bicarbonates. What are the phases? What are the regulatory part of that? Actually, small topic. This topic is very small. At the pancreas, which has been divided into the two part, means two as far as the physiological aspect and the working ability. Exocrine part and endocrine part. In the gastrointestinal system, what we are expected to learn or expected to note is the secretions and their role as exocrine effect. Exocrine. So the pancreatic secretion in the digestive gland or digestive system is with exocrine, means digestive enzymes and the secretion of the bicarbonate from the ductal epithelial cells. If you just join, those students who are late, please mute your mic just. Okay, if you have a query you can ask, no problem, but not during lecture. You will ask after also, or if you have a sudden question you ask it directly. Okay, so exocrine part of the pancreas, which does the effect on the digestive juices, means digestive secretions rather, So we have got the part with exocrine and endocrine. So endocrine secreting with the insulin along with the glucagon. So two important endocrine part which plays the role that we are going to learn in an endocrine system. This is what the location where we are going to have idea about this. Morning we are trying to learn this common bile duct. So this is with the coming from hepatic and hello, some students have mic is unmute actually. Sounds are coming. Please see if someone has kept the mic. Check your mic. Those students have keep mic open. Okay. So hepatic duct, which is coming from the liver hepatocytes, and the cystic duct, which is coming from the gallbladder. Then both together they join. Then it is called as with the common bile duct. Still someone. Please, someone has kept mic open. Please mute that. Thank you. So liver hepatocytes means that hepatic duct and the cystic duct, both together they join to form the common bile duct. This is the common bile duct which is coming as we have direction which is going through, and in between these coils of the intestine with the duodenum that join together with the pancreas. So the pancreatic duct along with the common bile duct, which is open within the name which is here as the duodenal papilla. So whether entering within the duodenum only. Most important. So duodenum is the part of the small intestine which virtually, that what we are knowing, that the components which are coming down as acidated chyme into the first part, that is duodenum only. So immediately there is a secretion. So almost physiologically we are knowing that purely acidic secretion which is going on, and that purely acidic secretion has to be reduced with its acidity. With some alkaline secretion has to be there. So bile is alkaline, which is liberated, and next to that immediately is the pancreatic. So both are trying to neutralize the effect of secretion of acid which is directly entered through. Otherwise, all of you are knowing it, if the acidated chyme directly enters into the part of the duodenum, so it will start to damage the intestines also. So basic role is to reduce that damage, prevent that damage by means of miracles under the development of human body or digestive system, where the digestive juices are so designed that the arrangement where we have the first alkaline, then we are starting with the acid, then we are starting again with the alkaline and alkaline and alkaline. So reducing or diluting the acid is the basic rule. So that's why this pancreatic juice plays the most important. So papilla of water. Where is the first important role of this papilla of water is to regulate, which is also called as the sphincter of Oddi. Now sphincter Oddi the sphincter Oddi, which is actually regulating the entry of the pancreatic as well as the bile which is coming from. So from the common bile duct, so we have got the bile duct from the liver which is coming on, which is joining here. So both with the pancreatic acinar cells here. So ductile epithelial cells and the acinar cells. So what we have learned with the exocrine part and endocrine part. So hormones which are liberated from the endocrine portion, which is the islets of Langerhans, almost, which is coming out from the part of the beta cell. Alpha cells which are liberating from the part of the Pancreatic secretion, which virtually drain into the duodenum. So the alkaline secretion has to pour with the two types or the two parts, where the acinar cells are there, which are secreting purely digestive enzyme, and the ductile epithelial cells, which secretes the tremendous amounts of the bicarbonate. So volume. If you are comparing about the pancreatic juice, approximately 800 ml to 1 liter per day. So this is what an average which is being secreted from the pancreatic secretions. So exocrine pancreas, which has been liberated, or which is playing the importance in the digestion. So we have got the headings over there. So I have gone through this directly. We have got the pancreatic secretion of that, which have a digestive enzymes responsible for proteins: trypsin, chymotrypsin, carboxyfolate peptidase, and elastase. The enzyme which is secreted for digestion of carbohydrate is pancreatic amylase, and the enzyme which is essential for digestion of fats, which is lipases, cholesterol esterase, phospholipase, and that cholipase, which is activated by trypsin, and already the bile salt activated by lipase. Now early morning we have learned in the morning lecture that the bile which is being liberated with the bile salts and bile pigments. So the bile salt which is essential for what? For the emulsification of the fat. So the fat digestion to form the miscellaneous complex or fat globules rather within the intestine after the effect of the bile along with pancreatic juice on the fats. So all these group of the enzymes which are responsible for the digestion, that is the exocrine pancreas, as it does this effect. Now see, just what in the gastric secretion in the morning, I am persistently talking with you the sentence I have used in the gastric secretion, that if the acid being liberated from the gastric mucosa, or it comes out within the canalicula or the lumen, can acid digest the gastric mucosa itself? That time the answer was, where the enzymes, rather than enzymes, the mucus which is secreted over there, that mucus plays a specific role, means they form a layer compound, total, mucin and mucus, whole within the gastric piece. So if the secretion goes on with the liberation of the from the parietal cells, they never damage to the gastric mucosa itself. This is what the interesting thing, and that's why the acid which never acts on gastric mucosa, because all of you are knowing it, that the mucosa, submucosa, all these layers and all, they have structure which are made up of the protein only. Lipoproteinous layers are there, means muscles are there, muscular layer with circular, longitudinal and all. And if acid which is being poured on the surface, or if acid that comes in direct contact with the mucosal lining, they will destruct it. So in a normal physiological process, that acid never damage the gas within a gastric pit to the any component of it. Same complex which is working with the enzymes which are liberated in the pancreas. When pancreas liberates these all enzymes, as we have mentioned, they are responsible for digestion of protein, responsible for digestion of carbohydrate, responsible for digestion of fats, And in fact, if all these group of the enzymes which are liberated, if they are remaining in an active stage, then what? No pancreas. No pancreas. I mean, that should not be the pancreatic secretion further. That means somehow these pancreatic secretions has to become activated. So I have written on this slide here, chyme, intestinal mucosa, enterokinase. Now this enterokinase enzyme, which is liberated from intestinal glands. Intestinal gland, as far as the location means, if they are located within duodenum, they will call it the duodenal mucosal glands, jejunum, ileum, so on. So all these inactive proenzymes, so they remain in inactive, trypsin, chymotrypsin, procarboxy polypeptidase, all these enzymes remains in inactive form. Necessity of that to prevent pancreatic digestion itself. So that's why the acinar cells of the pancreas, they're secreting it, but never digest the pancreas itself. Otherwise, it could have a proper digestion of that pancreas also. And pancreas will never left further for the secretion which is required. So enzymes to become active. So inactive form of the enzymes which are liberated within the duodenum, and the activation of the proenzymes which takes place in the lumen of the intestine. So it will depend on the intestinal state where the enzymes are liberated, means that after releasing through the sphincter oedi, means once that sphincter oedi opens and it liberates it, means it may be part of the bile along with the pancreatic juice, they will have activation. Okay. Now how it will get stimulated? Now students, these are the flow charts which are helpful to you, as if you have a less time when you are writing the answer in exam, this flow chart will help you a lot. Merely it gives more than expected marks at the last time if you are attempting any question and writing the answer. Such a flow chart are very helpful. So mind it well, this flow chart you doesn't require to have an explanation also. If you get the time, you explain it, otherwise just make out the flow chart, you will get the minimum marks actually, that what you require, rather within short span of time. Okay. So again come back to the intestinal mucosa, because once the sphincter oedi, which causes the relaxation, with the release of this all enzymes as a pancreatic juice directly, they will get the action of this enterokinase, means intestinal mucosa liberates the enzyme which is called the enterokinase, acts on all inactive enzymes to become active. So trypsinogen to trypsin, chymotrypsinogen to chymotrypsin, procarboxy polypeptidase to carboxy polypeptidase. So all three groups which needs to be get activated, and once they get activated, their activity starts. Means after a basic role for proper digestion of the acidated chyme along with the neutralization effect by the bicarbonates which are released. Same. Now that to relate with the acinar cells. Now this is with the ductile epithelial cells. What is the role of these ductile epithelial cells? They are releasing the bicarbonates secretion as to neutralize the acidic pH of the stomach, as the duodenum cannot resist that acidic pH. To block the peptic digestive activity totally with the gastric juices, these bicarbonates are functioning. They needs to provide the appropriate pH, and that pH, which is absolutely essential for the proper digestion. We have the proper digestion of the acidated chyme, which containing about the different required food stuff, has to be entered with this bicarb. Okay. So we have got the specific rollover with the functions of this, what is the role, why this bicarbonate has been, and from where it is being liberated. Okay. So next part of that is the phases. Already we are learning the phases and phases. We are learning gastric also. We are learning salivary secretion also. So if you remember, there are three phases: cephalic, gastric and intestinal. Now these three phases, where the percent of the secretions of the pancreatic juice as enzyme and to liberate further inside, the maximum percentage within the intestine when it with the local distension and the plexus has to stimulate. But anyhow, the secretions which being liberated within the paddock by means of stimulation from cephalic, 20% gastric, 10% intestinal, 70%. Means absolutely when food, means the desire, smell and the sight of the food, the hypothalamus centers and all, they needs to carry out the effect that I needs to eat this food, this type of the food. Immediately the secretion start, but actually we are not getting that particular type of the food in front of. Same thing happens in the salivary secretion, same thing happens in gastric with the different gastric gland, same thing which happens in intestinal secretions also, means purely intestinal juice also. And what we are learning is the pancreatic. So enzymatic activities for the liberation of the constituents of the pancreatic acinar cells or the ductal epithelial cells to release that secretion with respect to the feces, means depending on the food chain which is being present inside. So cephalic phase, all of you are knowing it. Again I am repeating, this could be the third time I think. Nervous signals from the brain that causes the secretion of stomach. Morning we have used that acetylcholine also. We have learned through the acetylcholine activity with the cholinergic fibers, secretion of which comes out as a bulk of the enzymes. And immediately the pancreas, which also release as a horizontal organ which is being located there. So secretion flows immediately from the pancreatic duct into the intestine because of only the small amounts of water and electrolytes are secreted at a time. So the enzyme has to be released within the duodenum with the relaxation of the sphincter. Next phase will be the gastric, means gastric phase of pancreatic secretion. Now see, where the gastric phase means actually now food has entered to the within a stomach, and we have mentioned that 5 to 10% has to be worked out for the pancreatic enzyme to act on further. So they will have the particular stimulation as the food being reaching within a gastric piece, but still the small amount that can be reached to the duodenum. Now you may have a question in your mind, why these phases are required? So significance is there, importance to the yeast phase, where the chyme which persistently has to flow after relaxation of the pyloric sphincter to get entered into the intestine. So the intestinal phase or intestine itself has to carry out the function in a proper way. They are stimulating it through the local reflexes, local you may call as Local factor as a distinction as the acidated chyme entered through, but the secretion has to persist. So first 10% has to be there. So 20 plus 10. So approximately 30% of the secretion which is there in the cephalic and the intestinal phase of the pancreatic secretion. With the large quantities of the bicarbonates are necessary for neutralizing effect. Next portion is the regulation. I told you this is easy path, just you go through as an overview, not in detail. Nervous and almost. So we have got nervous and hormonal everywhere. This is the acid. So stages which needs to cover up with the first acid from the stomach releases secreting from the wall of the duodenum. So that will depend on the fat or the amino acids which are being persistently being close to, or they were attached to, and then we need to carry out the attachment for. So release of each one with the cholecystokinin. Vagal stimulation is there for release of enzyme directly from the pancreas. So secretin and the cholecystokinin which absorbs into the bloodstream. Because the first sign or the first reflex which works is the vagal stimulation. Vagal stimulation, as far as the demand, where the enzymes are needed, needs to float. Apart from the hormones, which is the secretin, which basically supplies the maximum amount of the enzymes, along with the effect of cholecystokinin. Now this is a table, good table actually, shortcut, about hormones, secretin, release from the S cell of the duodenum. What is the role? Stimulus by acid or the acidic gastric juice. What it does actually? Effect on means that is the function of secretin. Helps in motility, where the mild effect on the gastrointestinal motility slightly reduced. Pancreatic secretion, most important, the secretion of bicarbonate. Basically, secretin, after release from the S cell of the duodenum, which directly acts on ductal epithelial cells of the pancreas to provide the more volume of the pancreatic juice to reduce the destruction of duodenal mucosa or the like. So the bicarbonate secretion by coming out. Cholecystokinin. Again being liberated from the duodenum and jejunum, eye cells. Digestive products for the fats, fatty acids, monoglycerides. So stimulant means if the intestinal contents have the maximum fat or maximum fatty acids or monoglycerides compound, it will stimulate to the cholecystokinin. So let's start with the contraction now. So in the morning also we have mentioned there cholecystokinin and gastrin. So strong contraction of the gallbladder, then inhibition of the stomach contraction and the pancreatic secretion of the enzyme. So stimulation is for the pancreatic secretion by means of release of the cholecystokinin. Now see, the stimulus from the duodenal lumen. It will comes out from the duodenal lumen. So it will act on the release. Actual role is to neutralize, and the second function is the digestion. So altogether they need to carry out the function under the influence of cholecystokinin and the secretin. So they will carry out the function with respect to increased secretion from the duodenal mucosa. Also the pancreatic duct which is being releasing it, and the cells continue further. So increased secretion of this bicarbonate actually neutralize the effect of acid liberated form. Then fats, proteins, products in the duodenum, means if the person has taken a more fatty diet, or if they have taken a more compounds of the fats or lipids, now they need to stimulate to increase cholecystokinin release from the duodenal mucosa. So the eye cells of the duodenum will immediately start the secretion. And that secretion has to regulate while acting on acinar cells, because the pancreatic cell which are liberating the secretion by means of the stimulation either from hormones or from the local distension. So acetylcholine and the cholecystokinin. Acetylcholine is a neurotransmitter. They are being released through the parasympathetic stimulation, right? Secretin, which is acting on the proteins and the duodenum. So they will have to a specific role for the digestion of proteins over there. So already we have discussed that the pancreatic secretion being produced in 3 phases: cephalic, parasympathetic stimulation by acetylcholine causes acinar cells to secrete the enzyme. Gastric phase has got same effect as that of the cephalic. Meaning whenever the cephalic phase is continuing with, they will allowing the acinar cells and the ductal epithelial cells to release within the duodenum. Now both the enzymes, or both the acinar cells and ductal epithelial cells in the intestinal phase, means actually the intestinal hormones secretin and cholecystokinin to increase release of the acidic chain and fats in the upper portion of the intestine. So respectively what happens, the components which are there in the part of the acidated chain has to be get neutralized, and that neutralization effect or neutralization effect will be depending on the phases. Cephalic, when it is with the gastric and the intestinal phase in combination, so that they have altogether activity. See, this is what I have covered with the pancreatic secretion. So morning we have gone through the gastric, then we have gone through the effect of secretion which has been out from gastric portion. Now we learn about some part in the small intestine. As all of you are knowing, saliva is the first. Saliva is the first. Gastric secretion is second. Then we have got the bile. Then we have got the pancreatic juice. And the last role of the intestinal phase. With the intestine, they have to carry out the effect of functioning with the secretion. So which is extending from the pyloric sphincter of the stomach to the ileocecal wall, where the maximum or the major site is for the digestion and absorption of the carbohydrates, proteins and fat. With respect to the number of enzymes which are located, or enzymes which are present in the part of the other juices, which are essential, means important, to carry out the process of digestion and absorption of different foodstuffs, means carbohydrates, proteins and fats. So here we have mentioned that length around 4 to 5 meters, 3 parts duodenum, ileum, just duodenum, jejunum and ileum. And these all 3 parts are secreting some sort of a secretion which is called as a intestinal juice or succus entericus. So the mucous membrane of the small intestine, which is covered by the intestinal villi, just like gastric pits, here are the villi. The projections which are existing in this with respect to the cells which is called as the enterocytes, enterocytes, enterocytes. That they will each enterocytes which are being present within the small intestine has to carry out the so electrolytes which gives with the hair-like projections called as the microvilli, and all it increases the surface area depending on the site of the intestines or the intestinal cells rather they has to release, along with simple tubular cells or the simple tubular glands which are lined by columnar cells in the intestine, which is being called as a crypts of lubricant. Crypts of lubricant. So columnar simple tubular glands lined by the columnar cells in the intestine called as crypts of lubricant. Sorry. So the columnar cells are within the active mitosis. The Brunner's gland also present. Brunner's gland name. Majority of the region which is carrying out is the Brunner's gland. Apart from the crypts of lubricant, which are secreting the intestinal juice called as succus entericus. Succus entericus. So enterocytes, intestinal or the intestinal hormones along with the juice which is responsible. Enterocytes mean digestive, liberated from globular cell, which is secreting purely mucus. Pancreatic cell, glycoproteins, immunoglobulins, lysozyme. The difference is almost the difference. The enterochromaffin cell, which serotonin, now that is serotonin is also essential for the secretion of the bicarbonates to work and to liberate itself. The gastrointestinal hormones are also there, so they will play the same role. Secreting cholecystokinin, then small amount of itself with the gastrointestinal peptides, gastric peptides rather. Again come back to the composition. Volume 1800 ml per day, alkaline, purely pH 8.3. Isotonic with the plasma. Composed of water about 98%, 2% remaining are the solid, divided into inorganic and organic. So it consisting of all the potassium, sodium, along with the calcium, the cations, and enhanced with the chlorides and bicarbonates. So the jejunal secretion and ileal secretion also, percentage of chlorides and the potassium is higher in jejunal secretion, and the renal, actually the ileal secretions which consisting of calcium max. So they have this directly with the inorganic salts to be get regulated here. Other constituents with proteins are there, mucins are there, albumin, immunoglobulins, numerous shells of the epithelial cells along with the intestinal mucosa. So the digestive enzymes present to straight about the epithelial cells simultaneously role in the digestion. They cover all activity of that. Now what are the functions of the succus entericus? Digestive function, by proteolytic enzyme, amylolytic, along with lipolytic enzymes. Protective means mucous membrane, mucus which protects the lining of the small intestine, preventing from the damage as the acidated chyme is. So there I have almost the Paneth cell also, intestinal gland, which defends, means playing the role in the defenses, means immunity or preventing the infection. Enterokinase, which is present in juice as the activates for the trypsinogen and trypsin. So this enterokinase is the most important one. Generally what we are knowing it with the activation only required with the all this group of activated enzymes means what? The intestinal cells are liberating it depending on the acidated chyme entry. It's not so with always the intestinal cells are secreting. Effective release from the duodenum and jejunum with trying to learn the concept of the cholecystokinin and secretin, and then activation. Already we have learned just before that the enzymes which are present in the location site or the cells which they are liberating, if we get some amount of the some amount of the particular stimuli for the activation of the, so that this is to convert into the active form of it. So digestive enzyme is the proteolytic, breakdown itself with the polypeptide into the amino acids, procarboxypolypeptidase, endopeptidases, dipeptidases. Enzyme actually acting on a nucleic acid, nucleases, nucleotides and nucleosides, almost all these three. What is the role? Why this is important? Intestinal lipase or the intestinal secretion which are coming out from the part of the intestinal cells, comparatively the intestinal lipase and the pancreatic lipase. If you are comparing about the functioning ability of the intestinal lipase is comparatively good. Other enzymes which are responsible for the carbohydrate, that all of you are knowing it, pass in 12th biochemistry. A specific 11th science also, I think few percent, I don't know exactly how much being there. So few enzymes which are there essentially for digestion of the carbohydrates. Few things, just like a salivary amylase there in the saliva. Apart from that, the persistent enzymes which are required are the maltase, sucrase, lactase, isomaltase, playing the role in the same process of digestion. Next part which comes, someone has kept the mic open. Regulation. All the secretions which are from the small intestine has to be get regulated by local neural and hormonal. Same points are there, same points. If you go through the topic of saliva, if you go through the topic of gastric secretion, bile, pancreatic, and last one is the succus entericus. All throughout they are carrying out the local stimuli which is being regulating. I am giving you these points as at least you could have some idea. It needs to have additional writing there. Local stimuli means local site, means exactly within the intestine. Presence of chyme in the intestine, which is increasing the secretion because of a local mind reflexes. So the intestine stimulates the secretion depending on the localized presence of. Second will be nerves, means vagal stimulation is there, secretions to be getting increased. Sympathetic stimulation is there to increase the secretion itself. So inhibition is there from the sympathetic. Parasympathetic has to work out with the secretion. Actual role is to prevent the loss of the fluid and electrolyte. But basically or directly they needs to get absorbed. So that proper absorption of the electrolytes under the influence of the intestinal secretion which regulates by local receptors. So we are not able to mean it with directly only the enzyme, only the digestive enzyme has to play the role. No. So the local receptors are there, hormones are there, which is called as the gastrointestinal hormones. Gastrin, intestinal secretion with the intestinal hormone, almost to provide it. So acetylcholine increases, which also inhibits noradrenaline, which inhibits the secretion. These are the homeostasis phenomena where they needs to regulate both. They are not allowing the excessive secretion. They are not allowing the excessive secretion also. They are trying to maintain the secretion in the specific format what they require. So the amount of the secretion from each juice where we needs to perceive about what will happen. The effect what we are learning itself by means of the secretions which has been poured through. Now the effect which work for the secretion of the different group of the enzymes liberating from either start from the saliva, you go through the end of the secretion up to the intestine. What for they are? They are for just the process of digestion, just the process of absorption. Everything which has been liberated from the part of the saliva down up to the intestinal, basic concept is to digest whatever the amount of the secretion, whatever the amount of the or the volume of the food, chyme which pass through. And virtually they will regulate the same component by liberating the enzymes either through the neural or by through the different stimuli or the localites. It merely not only they are controlling the secretion by means of amount or the volume of the food, but effects start when we have just with the thinking of the food. But as we have mentioned, in every phase is there, in the salivary, gastric, as well as in the part of the bile or succus entericus. We merely come to a specific point that the 10 to 30% of the secretions from the part of the gastrointestinal tract with respect to the glands has been poured out when we are just thinking of the particular type of the food. But if we are not taken it or not consumed, then the role of the digestive enzymes has to play. So virtually the enzymatic activity for the then to liberate more, then have additional secretion that continuously or persistently with respect to the localized site or the stimulation from where they liberate it. Means either in the gastric, means gastric glands, if you're talking about the hepatocytes in the bile, or the acinar or ductal epithelial cells. So I think with this I have completed your gastrointestinal system. I have taken fast, actually your weightage is slightly lower, means low weightage is there, few topics or, but I have elaborated maximum. In spite of that, if you have a query still, your queries are always welcome. Someone has asked me to share these PowerPoints also in another group. So I will try to share these PowerPoints also. But problem is, I need to get download from your my PC or laptop again to your WhatsApp group. So better those students want your PowerPoints, means this PowerPoint presentation. Better if you have given your mail ID to me, then it will be better for me to share you rather than just on a WhatsApp. Yes, I will try to share on WhatsApp also, but that points remains throughout for you. So I will upload few notes also. Notes means some highlight point, not in detail what I have written. Yes. So thanks a lot. If you have a query that you can ask me, no problem. So I am ending this session with this completion and the declaration that this system is being completed. Still, if you have any queries, your queries are always welcome.